You know, usually when we picture like advanced medical treatments, the image that jumps to mind is this incredibly controlled environment.
Oh, absolutely. Like a
Yeah.
pristine, completely sealed hospital operating room.
Right. You imagine heavy metal doors, filtered air systems, and a massive team monitoring just a million different fail safes.
Yeah, it creates this psychological safety net, I think. We just assume that level of environmental control is standard for, well, anything involving our bloodstream.
Exactly. But then, and this is what's wild, you step into the world of outpatient regenerative medicine, and that pristine bubble just vanishes.
Poof, gone.
You're just sitting in a standard clinic chair. A phlebotomist draws your blood, puts it into a machine buzzing right there on a normal counter, and maybe, I don't know, 30 minutes later, that processed blood is injected straight back into your knee, or your scalp, or your face.
Right, and the physical environment there is just entirely different, which fundamentally alters the mechanics of keeping that whole procedure sterile.
So we are jumping straight into the deep end of platelet-rich plasma therapy today, PRP.
It is everywhere right now.
It really is. People use it for orthopedic healing, wound care, skin rejuvenation. And the marketing pitch is brilliant because it sounds, invincible. It's your own blood, your own body's healing factor.
Right, what could go wrong?
Exactly. But the moment that blood leaves your vein and hits the air of a standard clinic room, we have a problem. How do we actually know it's safe to inject back into you? Like, what are the actual laboratory quality control measures keeping you from getting a massive joint infection when processing PRP in an outpatient setting?
Well, to figure that out, we're pulling from a really fascinating stack of sources for this one.
Yeah, some heavy reading.
Definitely. We've got FDA biological device approval summaries, the incredibly strict protocols from the AEB.
That's the Association for the Advancement of Blood and Biotherapies.
Exactly. Good catch. And we also looked at several cutting-edge medical journal studies that dive into microbial contamination risks, plus the wildly complex tech they're trying to use to detect those risks in real time.
So if we're going to look at how a clinic actually keeps your PRP safe, I feel like the most logical starting point is figuring out who is setting the rules for them.
Yeah, that's where things get interesting.
Because I dug into the FDA summaries, and there is a massive misconception here. You'll see clinics plastering FDA approved all over their websites for PRP therapy, but PRP itself is not an FDA approved drug or biologic.
Yeah, and that catches a lot of patients off guard. But the regulatory logic here is based on something called the same surgical procedure exception, which the FDA clarified back in 2017.
Right.
To understand this, you kind of have to think about what a pharmaceutical drug actually is, right? It's mass-produced in a factory, shipped globally, and designed to interact with millions of different immune systems.
Which obviously requires like decades of clinical trials.
Exactly. But PRP is autologous, meaning it's made entirely from your own biological material. You're literally just moving your own tissue from a vein in your arm to, say, a tendon in your ankle during a single uninterrupted visit.
So the FDA's stance is essentially, we regulate mass commerce, not the biological equivalent of moving furniture from your living room to your bedroom.
Huh. Yeah, that actually captures the spirit of it perfectly.
Yeah.
The FDA doesn't require pre-market approval for the plasma itself because it never enters commercial distribution.
Okay, but if the FDA is leaving the actual biological recipe up to the individual clinic, the only defense you have as a patient is the, Tupperware they store your blood in.
Right.
So the FDA must be heavily regulating the hardware, right? The centrifuges and the tubes.
Oh, they absolutely do. Devices like the Helium Float, the FG001, or the Illuminate PRP kits, those go through the FDA's 510K clearance process.
Okay.
The manufacturers have to rigorously prove that their centrifuges separate blood components accurately. Well, more importantly, that the tubes and kits themselves won't introduce any harm into the sample.
I was trying to map this onto something familiar, and it feels a lot like a restaurant kitchen.
Oh, how so?
Like the health inspector of the FDA comes in and certifies that the industrial oven is calibrated, the pots are, food-safe stainless steel, and the fridge stays cold.
Right.
But they aren't standing over the chef tasting the soup. They aren't approving the specific meal.
Exactly. And if the chef decides to just, leave the raw chicken on the counter for 4 hours, those food-safe pots don't matter at all.
Wow, yeah.
So this regulatory framework shifts a massive and often invisible burden of quality control directly onto the outpatient clinic. They become entirely responsible for implementing laboratory-grade daily protocols to prevent bacterial infections and to avoid pyrogens.
Actually, we should probably define that term quickly because pyrogens are a huge part of this story.
Yeah,
A
definitely.
pyrogen is essentially any substance that, if it gets introduced into your body, triggers a severe fever and just a massive inflammatory response.
Right. And you definitely do not want a massive inflammatory response raging inside a freshly injured joint you're trying to heal.
No, that sounds like a nightmare. So if the burden is on the clinic's kitchen, let's look at those pots and pans. How good is the hardware at keeping contaminants out?
Well, the manufacturing standards for those 510K cleared kits are intense. Before a single collection tube is allowed to be shipped to a clinic, the manufacturer has to prove it achieves a sterility assurance level of 10 to the -6.
Wait, meaning if you tested a million tubes, only one would have a single viable microorganism inside it.
Correct, 1 in a million. And proving that requires passing some serious ISO standards, specifically ISO 3004 and 19937.
So they don't just, like, zap them with radiation and call it good.
Not even close. I mean, they do sterilize them with gamma radiation or ethylene oxide gas, but they also have to prove that a sealed TRP kit will stay perfectly sterile, sitting on a sunny clinic shelf for up to 3 years.
3 years? How do they test that?
Through accelerated aging, they place the kits in these environmental chambers that blast them with extreme heat and humidity, where a few weeks simulate years of real-world degradation.
Oh.
Yeah, and then they test the seals to ensure absolutely no microscopic breaches occurred.
Honestly, that gives me a lot of confidence in the kit itself. But the moment the doctor rips open that 3-year tested seal and draws your blood, the path sort of splits, right?
It does.
Because based on the sources, clinics generally use one of two methods to prepare the PRP: closed systems and open systems.
Right, so a closed system is basically a commercial kit that acts as a contained loop.
Okay.
The blood is drawn into a sealed container, it goes into the centrifuge, and the final plasma is extracted with a syringe, all without a single drop of the blood ever being exposed to the ambient air of the room.
The blood never meets the clinic air. I mean, that sounds like the obvious choice for safety, but clinics still use open systems. Why?
Because an open system relies on standard test tubes and pipettes. After the blood spins in the centrifuge and separates into layers, a technician literally uncaps the tube, reaches in with a pipette, draws off the specific plasma layer they want, and transfers it to another tube.
So the blood actually physically interacts with the clinic environment?
I have to challenge the logic of that. If a closed system guarantees my blood never touches the room's air, why on earth would any doctor choose to uncap a tube of my blood in a standard room?
It really comes down to medical customization. Closed systems are rigid. The kit is designed to yield a very specific concentration of platelets, and, you get what you get.
Right.
But regenerative medicine is highly nuanced. If a physician is treating a chronic pendant issue, they might want leukocyte-rich PRP.
Meaning they want white blood cells in there.
Exactly. They specifically want to include a layer of white blood cells to kickstart a stalled healing process. But for, say, a facial aesthetic treatment, those same white blood cells might cause unwanted swelling, so they want pure PRP instead.
Oh, I see.
Yeah, so an open system allows a skilled technician to manually isolate the exact cellular layers they need for your specific injury.
Okay, I see the clinical benefit. But the risk of uncapping the tube just seems way too high. Do
Yeah.
they just wipe down the counter really well and hope for the best? Oh, definitely not.
Mhm.
I mean, if they are following proper laboratory quality control, they're doing much more than wiping down a counter. Safe open system preparation requires a laminar airflow hood.
What's that?
It's a specialized workbench that takes air through a HEPA filter and pushes it out in a heavy, continuous, unidirectional sheet.
Like an air curtain.
Exactly. It creates an invisible curtain of pure air that physically blows airborne dust and microbes away from the open blood tubes.
Okay, that's pretty cool.
Combine that with sterile gowns, masks, and strict aseptic pipetting techniques, and the risk drops dramatically. Plus, PRP actually has a fascinating biological failsafe built right in.
Right, the platelets themselves fight back.
They do. The high concentration of platelets and plasma components actually possess strong antimicrobial properties. They actively neutralize certain strains of bacteria.
So the fluid itself is defensive.
Highly defensive.
Defensive, but as we saw in the research, definitely not invincible. Which brings us to, honestly, the most startling part of this deep dive.
Yeah, this part is wild.
Even with an FDA-cleared device, a HEPA-filtered airflow hood, and a masked clinician, there are invisible threats that are shockingly hard to defeat, starting with the patient's own arm.
Right, because the human body is essentially a walking ecosystem. We are completely covered in microorganisms.
See, I just assumed the alcohol wipe took care of all that. But there's a study from the PMC journal in our stack that tracked microbial risks in PRP patients.
Yeah, the results were pretty eye-opening.
Very. The nurses aggressively disinfected the patient's arms with standard alcohol protocols. But when researchers swabbed the exact venipuncture site right before the needle went in, they found surviving microbes in 6 out of 10 patients.
60% of the time, the injection site was still colonized.
If the skin is still colonized and a hollow needle has to punch through that skin to reach the vein, isn't the needle just acting like an elevator, just carrying those surviving bacteria straight down into the sterile blood sample?
It is a massive vulnerability. The limitation of a surface alcohol wipe is that human skin isn't flat glass.
Yeah.
It's extremely porous.
Right.
It has deep hair follicles and sebaceous glands. Bacteria just retreat into those microscopic canyons where the alcohol literally evaporates before it can reach them.
Oh, that's slightly horrifying.
It is. So when the needle cores through the skin, it absolutely can pick up those deep-dwelling microbes.
So how on earth do we mitigate that? I mean, we can't autoclave a patient's arm.
No, we can't. So this is where outpatient clinics must borrow really stringent protocols from the AABB, the same rules used for massive blood banks.
Okay.
The primary defense against this skin flora infiltration is called the single uninterrupted venipuncture.
Meaning the phlebotomist can't, like, dig around, no poking, pulling back, and adjusting the angle.
Exactly. One perfectly clean slide into the vein. Because any repositioning basically acts like a microscopic plow, churning up more tissue and bacteria.
Wow.
But the truly ingenious mechanism is what happens immediately after the needle hits the vein. Modern best practice requires diverting or discarding the very first milliliter of drawn blood.
Wait, because that first milliliter washes the needle out.
Precisely. That initial burst of blood acts as a biological plug.
Oh, that makes so much sense.
Right. It catches the skin tissue and the surviving bacteria from the hair follicles that were trapped inside the hollow core of the needle. So by bleeding that first milliliter into a separate diversion pouch, you ensure that the subsequent flow of blood rushing into your primary sterile tube remains utterly pristine.
You essentially sacrifice the vanguard of the blood draw to protect the rest of the sample. That is brilliantly simple.
It elegantly solves a really complex biological problem. But bacteria aren't the only invisible threat we have to worry about here. We actually have to circle back to the plastics used in the clinic.
Ah! We need to talk about pyrogens again.
I do.
Right. So earlier you mentioned pyrogens cause terrible fevers, but if the sterilization process, the heat and the gamma rays used on those tubes, kills all the bacteria, why are we still worried about them? They're dead.
Because your immune system doesn't only react to live swimming bacteria.
Mm-hmm.
An endotoxin, which is a common type of pyrogen, is basically a shattered fragment of a dead bacterium cell wall.
Okay.
Even though the bacteria is completely dead and incapable of multiplying, if that jagged cellular debris gets injected into your joint, your white blood cells recognize the foreign structure. They sound the alarm, triggering a massive inflammatory response.
Wait, so the sterilization process actually creates the pyrogens by blowing the bacteria to pieces and then just leaving the toxic shrapnel behind in the tube.
That is exactly what happens. And this leads to a shocking reality regarding standard clinic equipment. Many clinics use everyday plastic vacutainer tubes, the ones containing the anticoagulant sodium heparin for PRP draws.
Okay.
But those standard plastic tubes have routinely tested positive for endotoxins.
Why would a medical manufacturer allow endotoxins in a blood tube?
Because those tubes are manufactured for diagnostic purposes. If you're just drawing blood to send to a lab to check a patient's cholesterol, a few microscopic fragments of dead bacteria in the plastic don't matter at all to the lab machine.
Oh, so the problem is when a clinic improperly uses those same diagnostic tubes for a therapeutic purpose, processing blood that will be injected back into a human being.
Exactly, because the immune system will definitely notice those dead bacteria fragments.
So what's the solution?
Which is why multiple medical task forces strongly advocate for clinics to use specialized glass vacutainers instead of standard plastics, because glass manufacturing can much more easily achieve a completely pyrogen-free state.
It is wild how a seemingly trivial choice between a plastic or glass tube can dictate whether a patient leaves with a healing knee or a severely inflamed one.
It really is.
Let's assume the clinic does everything right, okay? They use the glass tubes, they execute the biological plug diversion perfectly, and they utilize the laminar airflow hood. The centrifuge stops spinning, and the PRP is ready. This is where the tension really spikes for me.
Yeah, the problem of time.
Right. A pharmaceutical company can make a batch of vaccines, lock them in a quarantine warehouse for 14 days, and culture samples in a petri dish to guarantee nothing grows before shipping them.
Yep.
A PRP clinic does not have 14 days. I am sitting in the chair, the doctor has the syringe, the plasma is going into my knee in like 3 minutes. How does anyone actually verify it's clean right then and there?
Historically, they haven't verified it.
Wait, really?
Yeah, they've operated entirely on blind trust in their protocols. But the race to develop rapid microbial detection for autologous blood is one of the most intense frontiers in science right now.
So my first thought goes straight to PCR, polymerase chain reaction. We all got very familiar with it during COVID. It detects genetic material incredibly fast.
That-
Why can't we just drop a sample of the PRP into a PCR machine?
Well, the researchers in the PNC study attempted exactly that. But PCR proved to be catastrophically sensitive in all the wrong ways.
Oh,
PCR doesn't know the difference between a live, dangerous bacterium and a harmless, dead fragment of DNA just floating in the environment. So it triggered a massive number of false positives, which would force doctors to throw away perfectly good PRP.
okay, so it panics over ghosts.
Pretty much. And worse, it also suffers from false negatives.
Wait, it misses things too.
Yeah. Blood plasma is this thick, incredibly complex soup of heavy proteins, sticky clotting factors like fibrinogen, and dense cellular material. When scientists tried to extract bacterial DNA from that soup, the plasma proteins literally gummed up the chemical extraction process.
Oh, man.
The PCR completely missed dangerous bacteria like Cutibacterium acnes because the plasma itself shielded the bacteria from the test.
Okay, so if PCR gets blinded by the plasma proteins, what is the next best option for testing the blood while I wait?
The most promising technology right now is flow cytometry.
Okay, I read about this in the sources. It's essentially like a bouncer at a nightclub using a flashlight to check the ID of every single person walking single file through a narrow hallway.
That is a phenomenal analogy. Yeah, flow cytometry pushes the fluid sample through a microscopic channel so the cells pass by one.
Single file.
Exactly. And a laser hits every single cell. And sensors measure how that light scatters. The forward scatter tells the machine the exact size of the cell, and the side scatter reveals the internal granularity or, the complexity of the cell.
So it can look at the physical shape and basically say, that's a white blood cell, that's a platelet, and wait, that's a live bacterium?
Precisely. It is incredibly fast and can detect viable living bacteria within just a couple of hours. The sensitivity is getting down to 100 to 1,000 colony-forming units per milliliter.
Okay, let's translate that metric for a second. A colony-forming unit, or CFU, basically means one single bacterium capable of multiplying.
Right.
So if I have a milliliter of plasma, which is what, about a fifth of a teaspoon, the laser can spot it even if there are only a few hundred bacteria hiding in there?
Exactly. It's a massive leap forward. But it still suffers from blind spots because blood is just so messy. The researchers found that flow cytometry completely failed to distinguish a specific fungus Candida albicans from the natural cellular debris of the PRP.
Why? Do they look the same?
Well, when platelets clump together, which they naturally do to form clots, the resulting clump scatters the laser light in the exact same size and shape pattern as a yeast cell.
Ah, yeah.
Yeah, so the machine couldn't tell if it was looking at a healing clot or a dangerous fungal infection.
So even the lasers get confused. And flow cytometry only looks for physical, intact cells anyway. What about the pyrogens? The dead endotoxin fragments we talked about. How do you rapidly test if the plasma is going to cause a fever?
Good question.
Do they just, inject a tiny bit into a lab rat in the back room and wait to see if it gets hot?
We have definitely moved far beyond animal testing for this. To rapidly test for pyrogens without needing fresh human or animal blood, science uses the monocyte activation test, or ANT.
How does MAT replicate a human fever without a human?
They use cryopreserved human immune cells, specifically monocytes, which are kind of the sentinels of our immune system. Laboratories freeze vast, standardized batches of these cells.
Okay.
To test your PRP, they thaw a small amount of these immune cells and expose them to your plasma sample. If any pyrogens or endotoxins are present, those immune cells immediately wake up, attack, and release inflammatory chemical markers called cytokines.
And we can measure those.
Easily and rapidly. It completely removes the need to wait days for a bacterial culture to grow.
That's amazing. But even with MMAT for pyrogens and flow cytometry for live bacteria, we are still talking about an hour or two of processing time.
Yeah, that's true.
I'm still sitting in the clinic chair with a bad knee. The reality is they can't test my specific syringe of plasma right before they inject it.
No, they can't. Which brings us to the ultimate takeaway regarding safety. Because rapid testing is not yet instantaneous, outpatient clinics must rely entirely on standard operating procedures, or SOPs. These written rules are basically the only invisible shield protecting you.
And this is where the AABB mandates take center stage.
But exactly. The AABB rules leave absolutely no room for improvisation. They dictate the precise mathematical ratio of blood to anticoagulant to ensure the blood doesn't clot in the tube.
Wow.
They mandate 2 distinct patient identifiers on every single tube at every step of the transfer process to prevent catastrophic mix-ups. And they outline immediate, rigorous centrifuge cleaning protocols using hospital-grade biocides after every single spin.
They even dictate the exact temperature the room and the centrifuge should be. But I noticed in the sources that temperature control is actually the subject of a massive debate right now.
Oh yeah, big debate.
Because the AABB mandates storing the processed plasma between 20 and 24 degrees Celsius, so essentially room temperature. But there are dermatology studies arguing aggressively for a cold spin at 12 to 16 degrees Celsius. Why the fight over a few degrees?
It reveals how nuanced the biology really is. The AABB standard is based on classical blood banking, keeping cells alive and stable for transfusion.
Right.
But the dermatologists aren't just trying to keep platelets alive. They're trying to manage their behavior. Platelets are highly sensitive. At room temperature, they are warm and metabolically active. The sheer physical stress of spinning in a centrifuge at room temp can cause them to prematurely activate and release their payload of healing growth factors right there inside the plastic tube.
So the healing factors get dumped into the garbage instead of into your face?
Exactly. So the advocates for the cold spin argue that chilling the blood to 12 degrees essentially induces a temporary metabolic paralysis. It puts the cellular machinery to sleep.
Oh, that's clever.
Yeah, so the platelets survive the traumatic forces of the centrifuge without panicking and degranulating, ensuring they only wake up and release their growth factors after they are injected into the warm environment of your tissue.
It is incredible. The clinic isn't just managing safety, they have to manipulate the ambient temperature just to trick your cells into behaving.
And that level of precision is entirely in the hands of the clinician standing in front of you.
Which brings us to what this all actually means for you, the listener. Like, why should you care about flow cytometry, biological plugs, and cold spins? Because this intricate web of science is exactly why you cannot be a passive participant in regenerative medicine. Before anyone draws your blood, you have to ask the hard questions. You need to ask your provider. Are you using an FDA-cleared closed system? If you use an open system, where is your laminar airflow hood? Are you drawing with plastic diagnostic tubes or glass therapeutic tubes?
It's the most vital step a patient can take. You are delegating your biological safety to their in-house protocols.
Don't be afraid to be the patient who asks how the sausage is made, or in this case, how the blood is spun. Because the difference between a pristine biological treatment and a joint infection often comes down to an invisible curtain of HEPA-filtered air and a clinician willing to throw away the first milliliter of a blood draw.
The protocols are rigorous, but they only work if the humans in the room follow them flawlessly.
It's a fascinating look at the tightrope outpatient clinics walk every single day. But, looking at the trajectory of this rapid testing technology, it sparks an incredibly wild thought.
Oh yeah, how so?
Well, we've been talking about clinics, but if things like flow cytometry and monocyte activation tests eventually shrink down from clunky lab machines into fast, cheap, handheld devices,
Oh yeah.
right? We might not need the clinic at all. If we can generate a real-time digital sterility score for a blood sample in 3 minutes, we could safely perform advanced autologous regenerative medicine anywhere.
Anywhere.
Imagine processing and injecting healing stem cells and PRP in a dusty battlefield triage tent, or even on a deep space mission to Mars, completely independent of a sterile hospital environment, simply because a handheld laser confirms the plasma is clean.
Taking the safety of the operating room and putting it on a microchip, that would fundamentally redefine the boundaries of modern medicine.
Until that microchip arrives, make sure you know exactly what kind of kitchen your doctor is running. Thanks for joining us on this deep dive.